Good evening, everyone. First of all, again, thanks to the organizer for having me here. While you are enjoying your dinner, let me briefly introduce myself. Right, I am from the Department of Tissue Engineering and Regenerative Medicine based in University Kebangsaan, Malaysia. I've been doing this for the past 20 years. And today, I would like to share, I hope it's not too technical. I'll try not to be too technical. I'd like to share with you the topic on healthy aging and its association with stem cells. Some of these terms that you may come across, but I will explain it as we go along. Okay, stem cells, telomere, telomerase, mitochondria, and so forth. Okay, first of all, I need to make this disclaimer. So the views I express in my presentation are my own and do not necessarily represent the views of UKM. And additionally, I do not work for BioLife and I do not represent them either. Okay, so and this is my team in tissue engineering department. This has been taken long ago when I was the head of department, and now we have a younger head, a very dynamic head of department. The Department of Tissue Engineering is on the top floor of the hospital, UKM Hospital in Cheras. If you have ever been to Cheras, Kuala Lumpur, you'll find the complex, the medical complex of UKM there. So this is the entire complex, and the complex comprised of even the children's hospital and the preclinical, the faculty of medicine where we train our undergraduates. All right, so let me share with you my journey. I started my research on cancer. And what I've learned from my cancer study is, I summarize in these 3 points, that cancer cells can live forever, and given the right condition, they will continue to grow. And one of the ways of most cancers, not all, but most cancers, overcome this problem of growing old. Cells do grow old, but cancer cells don't grow old, is they can turn on an enzyme, a protein called telomerase, and this telomerase allows them to keep the chromosome, which is the end of their chromosome called telomere, in a length that is maintained. It's short, but it is maintained. So don't worry, I will explain to you how I apply this in my study now. So that was, it fascinated me that cancer can grow outside the body and in the dish forever. And in my PhD, I went into stem cell and regenerative medicine. And when I embarked on this journey, it was at the height of tissue engineering. You would see pictures like this at that time. It was very new. It was, I think it shook the world, because now the idea of growing the entire tissue outside your body or in an animal becomes a reality, and to some it's a horror, right? Now seeing a human ear behind a mouse, right? But that becomes a dream of many who may be waiting for a tissue transplantation or for an organ to be transplanted, right? So I summarized the approach of tissue engineering in this slide. So the idea was, if we can take a piece of tissue from you, we can grow your cells outside your body, and by adding the cells into a material like a house, a housing, then it would give it shape and dimension, like your tissue, right? Yeah, like your a piece of skin, or like your piece of part of your organs, right? So the housing is what we call scaffold, and then we will mature it outside your body by giving it some stimulus, like biochemical stimulus. It could be some mechanical stimulus or electrical stimulus, and that allows the tissue to mature enough to be functional, so that we can transplant it back to the patient. So this was the simplistic approach of tissue engineering. I think I can say it is still what we are doing now. To give you an idea, what it looks like when a cell is taken out of your body, you see we can control it in many ways. We can make it grow, make it migrate or move in a way that we want. Meaning, in this video, you can see the cells are moving in one direction because we want it to move in that direction. And we can give it cues to differentiate, meaning turn into a cell of different type. Right? So, I just want to impress upon you that outside the body, these cells can be manipulated, meaning we can control them by adjusting the environment that they lived in. The video below are cells that are grown on 3D scaffolds, and according to the different types of scaffold, they will turn into different types of cells. To give you a snapshot of the kind of tissues that we can engineer, so we have successfully engineered cornea from the eye. For the eye, we engineered nerve, bladder, bones. Okay, we even try with injectable bones, where we can inject into a bone defect, a bone gap that, if you leave it alone, will never unite. Right? So these are critical bone gap that you can even inject cells and the material that I mentioned into that gap and allow it to grow. So this is a reality, and we have shown it in many of our animal models. And to make it more realistic, in a clinical scenario, you may come with a patient may come with a fracture, a comminuted fracture like this. So in this case, the surgeon will usually take away the bone, the fractured bone, and there would be a bone gap. There would be a gap so big that often you would have to be on fixators, right? And if it's small enough, probably they would allow it to slowly grow back, or use a technique called something like bone transport to slowly, but it's a very lengthy process and painful process, and many a times you may get infection. One more, if you are older, it may not if ever really unite. So by having this kind of models, we are able to the 3D scan we are able to customize the tissue that we engineer according to the size and dimension of the bone defect. So this was what we did. So give you an example, we did this in our lab, and this is a bone that we customized to our sheep, and 3 months later, by injecting the stem cells from the sheep. So these are mesenchymal stromal cells that we sometimes use interchangeably, stromal cells or stem cells, more popularly known. But in short, we call them MSCs. These are cells that you can be derived from your bone marrow or fat. In this case, it's from the sheep bone marrow, and we seeded these cells onto material. In this case, this is a ceramic material, very close to your bone, and we implanted it into the bone gap. And 3 months later, new bones were formed, and the bone were united. So it was not easy to do this in a sheep, because the sheep will never rest in bed. The very next day, or even after they've woken up from the anesthetic drugs, they start to walk. They even try to walk on their 2 hind legs, which is jump on the 2 hind legs, which usually we won't let patients do, right? So it was very challenging. But anyhow, we were able to unite and regrow the bone. And another example, knee cartilage. Many old people will have this experience of osteoarthritis of the knee. So with the cartilage defect, in the sheep that we created, we have also injected mesenchymal stromal cells, and we have shown that we are able to regenerate cartilage. Maybe to give you a better picture, this is another sheep study that we embark on. We surgically remove the ligament that holds your knee together. We even remove the meniscus, and we allow the animal to run around for 6 weeks until it develops osteoarthritis. And with that, we again injected MSCs, and here we track the cells. The red color are the cells that we have injected. So the important key here is these cells are from the animal. The animal own MSCs. So I want to highlight this because there are different strategies out there using your own MSCs, your own cells, or cells from others. Right? So in this case, they are the sheep's own cells, and we track them. And you could see 3 months later, the new cartilage that was formed is a very nice layer. You could see a thick layer. The red color is because we have stained them, we have tracked them, and it grew into a nice layer of smooth cartilage. And it expresses collagen type 2 and SOX9, which is the biomarker of the kind of cartilage that you want, the high-lying cartilage, not your scar tissue usually repairs such defect. And so I hope you are convinced that the stem cells that we injected was able to regrow the lost cartilage. So we went on. I will give you 2 examples. There are a few more examples, but I will give 2 examples where from the animal study that was found successful and promising, that we brought it to clinical trial. So the first one was an engineered skin. Again, we use cells from the patient. So you can see in the picture that we have a piece of skin from the patient. And usually patients that need skin graft would be because either they have a very large wound or they have a chronic ulcer that would not heal, right? That a wound will never heal for over a long time, and so we have to make an engineered skin. The first study was in 2004. This was we did not start the study. This was a request that came out of the blue because it was a girl that was referred to us with a 34% burn, and she was already in sepsis. So when she was referred to us, we thought she might not even make it. So after stabilizing her, there was a request from the plastic surgeon. They were running out of skin to be grafted. So in order to graft, you need to get skin from a healthy part of her body, right? So there were not enough healthy skin to cover. They were even contemplating using the skin from the scalp. So for that, they pleaded us to make this engineered skin, and we made pieces of engineered skin for this patient. In fact, it was when you're young, your healing is also much faster. So you can see actually it was very extensive burns. So we implanted initially 6, eventually we actually made more. I think we totally implanted like 10 pieces of skin on her. And this is 2.5 months later. And in this case, because we couldn't make enough also, at the same time she had skin graft. And for those of you who have seen a split skin graft, this is how it would have looked like. You can see part of the skin that looks like crumple and a bit like jigsaw puzzle. That's the skin graft, because those are mature skin that you transplant. But the pinky one are the ones that we make for them. So because that's new skin, like almost like baby skin. And to impress you further, the skin graft that you implant tend to contract, and usually causes a lot of problem as well in your movement. All right, so with that confidence, we went on to do a proper clinical trial on more patients. So this is another burn patient. Just to impress upon you that it's not just the skin that we are dealing with. We are dealing with psychological issues. So in this case, patient who have large burn issue and non-healing, doesn't heal for a long time, actually develop psychological problems. So that's why sometimes it is even longer. And this is a traumatic and accident wound that we have treated. Another case of trauma. So I'll just quickly, I'm sure you don't want to see this very gross, but this I would like to point to you is a diabetic patient. And we were able to, you can see from day 0 where I can see the bone and the ligament and tendon to day 90, and it was healing. Yeah, and eventually at this point, the doctor felt that he can quickly make a split skin graft and cover, because we didn't want to make more skin and let him wait. And another one is bone. So this is a direct product of my PhD study. Again, a case of bone infection, and started with a bone injury and then bone infection. The patient had grafted, from hip bone, grafted on the lost bone, but it did not unite properly, you could see. And the patient was wheelchair bound for more than a year. Very young, 20 over years old, motor vehicle accident. And with that, again, we make bone from 3D printing. And we assemble it together and grow the cells in our laboratory, which is a good manufacturing facility. And you could see me, growing the cells in the lab. And after probably around 3 weeks, we were able to implant on the patient. And this is a picture of the patient now walking. So, and eventually he actually got married, and now he has a daughter. So we just met a year ago, and this was 5-6 years down the line. And it shows that it is a safe and efficient treatment. If you are new to this, I think it is good that you understand that sometimes what they call stem cells are, they generally call everything stem cells, but some are real stem cells, some are not really stem cells. And there are also stem cells that are controversial, like, we get it from embryo. A lot of people have issues with that. But frankly, we do work on that as well on a research basis. And there's also stem cells that you can generate from skin. I can turn it from a skin cells. We can turn it into what we call an induced pluripotent stem cells, and that's also what we do in our lab. So fear not. Even now, if you feel that you have limited cells or stem cells in the body, we can actually make them in the lab. And the main sources of the exist now. What is the kind of stem cell sources that is still in your body? We can get it from bone marrow. I mentioned it earlier. And we can get it from the embryo. I also mentioned it from the embryo. And we can also get it from other parts of your body, for example, like blood. So many people would confuse like blood stem cells with other types of stem cells. So these are stem cells that can, when we say blood stem cells, these are stem cells that will turn into blood cells. So we say skin stem cells, I would say they are more like progenitor cells, that the earlier form of stem cells of the skin that will eventually turn only into skin cells. And we have the MSCs that we mentioned earlier, and this is very interesting because it has a lot of therapeutic use, and we can derive it from bone marrow, from fat, from tooth, from the placenta, and very interestingly, we can get it from thrown away discarded tissues like the cord, the umbilical cord, and the placenta is full of MSCs, and even from apheresis, procedure. So these are all the sources of MSCs that we can get from you now. Okay. And to give you an impression how the cells grow outside your body, just now I show you how they can grow in unidirectional. This is a picture of how they close a gap. But you can see different cells grow at different rate. Of course, when you grow old, your cells are going to move slowly and they are going to proliferate slowly, and they also do not function that well. So I just want to impress upon you, your cells in your body do grow old. And when we take out from your body, not like cancer cells, they can't grow forever, they will age. So that you understand why your cells age. So your cells age because every time your cells duplicate, because your cells will grow, right? And every time it grows, it will shorten. The chromosome, what shortens is the chromosome, where it houses your DNA, your genes, okay? It will shorten. And of course, if it shortens every time, it will come to a point that some of your genes might be affected. So when that happens, your cells will die, okay? They will go into apoptosis. All right, so this is why your cells will never grow forever. So I give this example like, because this is like photocopying. Your cells need to photocopy the DNA, and every time when it photocopy, there is one part of it that it cannot photocopy. So that part will become shorter, and the next time it photocopy, also because maybe that part is the part where the protein that photocopies your DNA will have to park itself. So every time it parks there, it will not photocopy there. So every time it will become shorter and shorter. All right. So from the telomere length, we can actually predict how old the cells is. So similarly, if I can get your blood, we can also almost predict the lifespan that you have, how much your cells have already grown. This is just to show how much we can predict from the length of your telomere of your cells. All right, so I will introduce another thing that we in our research group is interested in. So besides looking at the telomere, we are also looking at what also besides that telomere that predicts your age, there is also something called epigenetic clock. And so epigenetic is, we have genes. And many a times, most of us think that, with that set of genes, you are either doomed because you have certain genes inherited, if that genes predict that in a certain way that you may have disease. But on top of your genes, actually there's another thing that controls it. It is whether your genes get expressed or do not get expressed. So that is called epigenetics. So that's what we're really happy, because that is something that we can change. So we may not be able to change your genes, right? You say that I inherited this from my mother, my father, my ancestors. We can't change that because it's throughout your body, but we can change the epigenetics, what controls the genes. And guess what? That these epigenetic changes is programmed in a way that we can predict how old you are just looking at what is being expressed and not expressed in what kind of epigenetic changes is on your chromosome or DNA. Okay, so that is called the epigenetic clock. This is a picture of young cells, MSCs, young MSCs versus old MSCs. And the blue color is a kind of enzyme that accumulates in their body because they are accumulating a lot of waste. So when we grow old, we tend to accumulate a lot of toxin. So similarly in the cells, it's so much that they cannot get rid of the toxin. So let's remind ourselves we have to detoxicate. As we grow old, we should not accumulate so much toxin. And when we have a lot of toxin, not only we are intoxicated, we sort of become inflamed. We secrete things that are toxic, and it affects the neighboring cells. So we become inflamed. So these are called senescent. We call it SSXP, meaning senescent associated secreted proteins. So I always remind myself, when we are old, we should not be inflamed, create a lot of toxin and become toxic to others around us. Okay. This accumulated toxin in your body, a lot of old cells that are staying in your body, and they become toxicated, and they secrete these proteins and affect neighboring cells, and it causes your body to be chronically inflamed. And so this low-grade chronic inflammation is one of the marker or one of the signature of an aged person. Yeah. So this kind of accumulation of toxic, it's also what affects the epigenetic. Just now I said the things that regulate your genes. Okay, so it will cause instability. So before that, things that are supposed to be expressed now don't get expressed. Things that are supposed to be expressed don't get expressed. So they become havoc, I would say. Okay, in an inflamed condition, and especially in aged cells when we study them in the outside your body. And so I just want to, just now we saw how telomere can predict your age. That's just the end of the chromosome, right? And we have a way to elongate, I will show you later. But these epigenetic changes even predict it even more accurately. So I again like to impress upon you, these epigenetic changes is, to me, is what drives aging. Alright, so I did say that, we can find a way to elongate the telomere. So we have found a way to overcome that. Of course, we can introduce telomerase. We can turn on the telomerase just like cancer cells. We can turn it on, and we can maintain your telomere length and allow your cells to grow longer. And this is what we have done in the lab. And we show just a few patients here that when you grow old, your telomerase activity drops. And we found that in men, maybe 60 years old, and in female, around 40 years old, we don't find telomerase activity anymore. Okay, and I said that we are able to activate the telomerase in these cells and allow it to grow. So within the cells that we will take up from the patient, we are able to turn it into a young stem cells before giving it to the patient. That was the objective, because no point giving you your old cells. Since we are able to manipulate these cells, not only we want to turn it to the cells that we want, we also want to turn it into a younger cells before we give it back to you. So that was what we have done so far. Okay, and another thing that we try to control is before giving the cells back to you, we are able to make it more energized by increasing its mitochondria. So we give an example here. Mitochondria is the tiny component inside your cell that give you energy. Okay, so the cells like heart has got a lot of this mitochondria because they need a lot of energy. So this is how it looks like. It looks like a worm, right? And we are able to induce your cells to create or to form a lot of this mitochondria before giving back to you. And why do we do this? I will show you in the next picture, because we have shown that if we inject these cells or we culture these cells along with cells that are dying. So the cells that we have induced a lot of energy, we create let it form a lot of mitochondria, very energy driven. That is the cell in red color. And the mitochondria, it has been stained with red. You can see also red color. And the green ones are heart cells that are dying because we starve them and we don't give it enough oxygen. And so when we culture these two cells together, so the red one is the stem cells, the MSCs that have been induced to form a lot of energy. What happens is the stem cells transport the mitochondria to the green cells naturally. Okay, so this, I always use this analogy on my students. I said even cells know how to do that. When they are full of energy, they are able to rescue the cells that are dying. And so we believe that by infusing these cells, it will rescue a lot of your dying cells in the body. I couldn't point to you, but if you see a very long hand in that picture, that very long hand, I think is very clear in that picture. The stem cells extend its hand to the other cell and transport the mitochondria to the other cells. Okay, so very quickly, this is again, but this was not taken by my lab, and in other labs they have also shown similar findings that the mitochondria gets transport from a more high energy cells to a dying cell. Okay, I will try to speed things up. And so we are all in this Malaysian demographics. We are all growing old, and by 2050, 1 in 6 people in worldwide will be over 60, and in Malaysia by 2030, 15% will be over 60. So we are slowly going towards an aging nation. And these are all the hallmarks of aging. I think Prof. Su will explain a lot about this. I wouldn't want to go into this. But of all the aging hallmarks of aging, I've mentioned telomere shortening. I've mentioned that your stem cells are growing old. I mentioned your mitochondria, no energy, cannot make enough energy. That's why you are also lethargic. All right, so these are epigenetics. These are the few key that our lab is looking at changing, and hopefully we can do something for longevity. And so what we really aim is not just long life, but we want you to be healthy. So before we can have a proven strategy for all of you, because all of what I've shown to you are promises that we cannot deliver yet now, because we still have to do a lot of research. But what has been proven here is this has been shown to help in increasing longevity. For example, caloric restriction. Therefore, you see most people try to eat less. Okay, get nutrition food, but eat less. Fasting, because all that has to do with reduced oxidative stress. Less stress, less inflammation, less epigenetic changes. Remember, right? So it will slow down your aging process, and it will also increase mitochondrial health, because it allows your cells to repair itself, not busy metabolizing. And then exercise and sleep, of course, enhance repair mechanism. And senolytics and probiotics. Senolytics is how certain drugs or certain chemicals can get rid of the old cells. But basically, I prefer not getting rid of the old cells. I prefer renewing the old cells. This is almost like saying that our society has got a lot of old people, we have to get rid of them, right? But I guess more important is let our old people become young again, right? I think that's better. And probiotics is just the gut, and it has found that the healthy gut can also reduce inflammation. So those are proven. So our goal is to have healthy lives, healthy span versus just long life. And with that, I want you to know that you are in control of epigenetics as well, because we have found a lot of environmental factors can affect your epigenetics health. For example, exercise, your diet, and even socialization has shown to affect your epigenetics. Alright, so all this can turn on and turn off your genes in a positive manner. So these are things that you can control. So I think with that, I'd like to conclude that, of all the strategies that you are trying to find out what can work for you, please base it on there is logic, there is real proof, there is publication on it, there's literature, and there are clinical trials, which is actual proof. Okay, so with that, I think I thank you for your attention.